infectious agents, internalize them, and destroy them. For this purpose, they are strategically placed where they were encountered such particles. For example, the Kupffer cells of the liver line the sinusoids along which blood flows, while the synovial A cells line the synovial cavity. Blood cells belonging to this lineage are called monocytes. In time, these migrates from the blood into the tissues where they develop into tissue macrophages. These cells are very effective at presenting antigens to T lymphocytes. The mononuclear phagocyte system has main functions two main functions which result the activities of two different types of bone marrow derived cells. First is professional phagocytic macrophages whose main role is to remove particulate antigens. Second is antigen presenting cells, APCs, whose role is to take off, process, and present antigenic peptides to T cells. Second is polymorphonuclear phagocytes. These have a lobed, irregularly shaped nucleus. They are classified into neutrophils, basophils, and eosinophils on the basis of how their cytoplasmic granules stain with acidic and basic dyes. The three types of cells have distinct effector functions. Now let's know about neutrophils. Most numerous are the neutrophils, also called PMNs, polymorphonuclear neutrophils, which constitute the majority of leukocytes in the bloodstream, around 60 to 70% in adults. They develop from the same early precursors as monocytes and macrophages. Like monocytes, they migrate into tissues, particularly at sites of inflammation. Neutrophils are released from the bone marrow at a rate of around 7 million per minute. They are short lived, that means 2 to 3 days, relative to monocytes or macrophages, which may live for months or years. Like monocytes, PMNS adheres to endothelial cells lining the blood vessels, marginate and extravasate by squeezing between the endothelial cells to leave the circulation. This process is known as diapedesis. Adhesion is mediated by receptors on the granulocytes and ligands on the endothelial cells and is promoted by chemoattractants such as interleukins 8. Neutrophils comprise over 95% of the circulating granulocytes. They have a characteristic multi-lobed nucleus and are 10 to 20 micrometer in diameter. Chemotactic agents for neutrophils include protein fragments released when complement is activated. Example, C5a factors derived from the fibrinolytic and kinin systems. The products of other leukocytes and platelets and the products of certain bacteria, chemotactic stimuli result in neutrophil margination and diapedesis. Neutrophils have a large arsenal of antibiotic proteins stored in two main types of granules. The primary azurophilic granules are lysosomes containing acid hydrolase, myeloperoxidase, and muramidas. The secondary granules contain lactoferrin and lysozyme. In addition to these enzymes and to lactoferrin, the granules also contain the antibiotic proteins defensins, granules, saprocidins, cathelicidins, and bacterial permeability inducing protein, that is BPI. Ingested organisms are contained within vacuoles termed phagosomes, which fuse with the lysosomes to form phagolysosomes. Extracellular release of granules and cytotoxic substances by neutrophils can also occur when they are activated by immune complexes through their ACY receptors. This may be an important pathogenetic mechanism in immune complex diseases. Now let's know about eosinophils. Human blood eosinophils usually have a bilobed nucleus and many cytoplasmic granules that stain with acidic dyes. Example eosin. They comprise 2 to 5% of blood leukocytes in healthy non-allergic individuals. Although not their primary function, they appear to be capable of phagocytosing and killing ingested microorganisms. The granules in mature eosinophils are membrane-bound organelles with crystalloid cores that differ in electron density from the surrounding matrix. Certain stimuli will cause eosinophils to degranulate. Degranulation involves fusion of the intracellular granules with the plasma membrane and release of the granule contents into the extracellular environment. This type of reaction is the only way that these cells can use their granule armaments against large pathogens which cannot be phagocytosed. Eosinophils are thought to play a specialized role in immunity to parasitic worms. Using this mechanism, eosinophils also released also release histamines and aryl sulfatase which inactivate the mast cell products histamine and some of the leukotrienes. The effect of the eosinophil factors is thus to dampen down the inflammatory response and reduce granulocyte migration into the site of invasion. Now, let's know about lymphocytes. Large numbers of lymphocytes are produced daily in the primary or certain lymphoid organs, that is thymus and postnatal bone marrow. Some of these cells migrate via the circulation into the secondary lymphoid tissues, that is spleen, lymph nodes and mucosa associated lymphoid tissues. The average human adult has about 2 into 10 to the power 12 lymphoid cells and the lymphoid tissues as a whole represents about 2% of the total body weight. Lymphoid cells account for about 20% of the leukocytes in the adult circulation. Many mature lymphoid cells are long-lived and persist as memory cells for many years. Two distinct morphological types are seen in the circulation as determined by light microscopy and a hematological stain such as GIMSA. The first type is relatively small, is typically agranular and has a high N is to C nitrogen is to carbon ratio. The second type is larger, has a lower nitrogen is to carbon ratio, contains cytoplasmic azurophilic granules and is known as the large granular lymphocyte, in short LGL. Lymphocytes occur as two major types, that is B cells and T cells, which are responsible for specific recognition of antigens. Hence, they initiate adaptive immune responses. All lymphocytes are derived from bone marrow stem cells, but T lymphocytes then develop in the thymus, while B lymphocytes develop in the bone marrow in adult mammals. Now, let's know about the B cells. About 5 to 15% of the circulating lymphoid pool are B cells defined by the presence of surface immunoglobulin. Each B cells is genetically programmed to encode a surface receptor specific for a particular antigen. Having recognized its specific antigen, the B cells multiply and differentiate into plasma cells, which produce large amounts of the receptor molecule in a soluble form that can be secreted. This is known as antibody. These antibody molecules are large glycoproteins found in the blood and tissue fluids. Because they are virtually identical to the original receptor molecule, they bind to the antigen that initially activated the B cells. The majority of human B cells in peripheral blood express two immunoglobulin isotypes on their surface, IgM and IgD. On any B cell, the antigen binding sites of these isotypes are identical. Fewer than 10% of the B cells in the circulation express IgG, IgA or IgE, although these are present in larger numbers in specific locations of the body. Example, IgA barriering cells in the intestinal mucosa, immunoglobulin associated with other molecules on the B cell surface forms the B cell antigen receptor complex, in short BCR. These accessory molecules consist of disulfide bonded heterodimers of Ig alpha, that is CD79A, and Ig beta, that is CD79B. The heterodimers interact with the transmembrane segments of the immunoglobulin receptor, and like the separate molecular components of the TCR or CD3 complex are involved in circular activation. B cell differentiation leads to the formation of plasma cells and memory cells. Following beta cell activation may beta cell. B cells blast mature into AFCS, which progress in vivo to terminally differentiated plasma cells. Some B blasts do not develop rough endoplasmic reticulum cisternae. These cells are found in germinal centers and are named follicle center cells or centrocytes. Under light microscopy, the cytoplasm of the plasma cells is basophilic. This is due to the large amount of RNA being utilized for antibody synthesis in the rough endoplasmic reticulum. At the ultrastructural level, the rough endoplasmic reticulum can often be seen in parallel arrays. Plasma cells are infrequent in the blood, comprising less than 0.1% of circulating lymphocytes. They are normally restricted to secondary lymphoid organs and tissues but are also abundant in the bone marrow. Antibodies produced by a single plasma cell are of one specificity and immunoglobulin class. Immunoglobulins can be visualized in plasma cell cytoplasm by staining with fluorochrome labeled specific antibodies. Many plasma cells have a short lifespan, surviving surviving for a few days and dying by apoptosis, whereas a subset of plasma cells with a long lifespan, that means months, has been recently described in bone marrow. We studied. Now let's know about the T cells. T lymphocytes are produced in the thymus from circulating T cell precursors derived from the hemopoietic stem cells in the bone marrow. These T cell precursors differentiate within the thymus into mature T cells. Precursors differentiate within the thymus into mature T cell expressing molecules important to T cell function, such as the T cell receptor CD4 and CD8. The thymus thus generates large number of functionally mature antigen-specific T cells which then migrate into the peripheral lymphoid tissues to mediate protection against invading microbes. There are several different types of T cells and they have a variety of functions. T helper type 1 cells, that is Th1 cells. One group interacts with mononuclear phagocytes and helps them destroy intracellular pathogens. They are called type 1 T helper cells or Th1 cells. T helper type 2 cells. Another group interacts with B cells and helps them to divide, differentiate and make antibody. These are the Th2 cells. Now, let's know about T cytotoxic cells. The third group of T cells is responsible for the destruction of host cells which have become infected by viruses or other intracellular pathogens. This kind of action is called cytotoxicity and these T cells are hence T cytotoxic cells. Cytotoxic T cells, also called killer T cells, destroy cells displaying a foreign epitope bound to an MHC protein. The binding of this foreign motif to T cell receptor causes the cell to secrete granules containing porphyrin. This 70 kD protein lyses the target cell by polymerizing to form transmembrane pores 100 angstrom wide. Perform is similar to C9. The final component is in the complement cascade. In every case, the T cell recognize antigens but only when they are represented on the surface of the other cell by so-called major histocompatibility complex, that is MSC molecules. They use a specific receptor to this termed the T cell antigen receptor, in short TCR. This is related both in function and structure to the surface antibody which B cells use as their antigen receptor. However, they differ from antibodies in several ways. T cell receptors are not secreted. The immune response mounted by T cells is triggered by receptors located on their surface. In contrast, the humoral immune response is mediated by soluble antibody molecules. T cell receptors do not recognize isolated foreign molecules. The foreign unit must be located on the surface of a cell. Specifically, the foreign molecule must be presented to the T cell by a particular membrane protein, one encoded by the major histocompatibility complex. Antibody recognize foreign molecules in solution and one membranes irrespective of their context. Most T cell receptors recognize relatively short peptides less than about 10 residues in length. These epitopes typically do not have a well-defined structure in solution. The conformational determinants detected by antibodies, on the other hand, consist of residues that are brought together by the folding of a long polypeptide chain. The essence antibodies recognize native macromolecules, whereas T cell receptors recognize fragments derived from them. T cells generate their effects either by releasing soluble proteins called cytokines which signal to other cells or by direct cells to cell interactions. Now let's know about T suppressor cells. Although there is clear evidence for the existence of antigen specific suppressor T cells, it is unlikely that they represent a functionally separate T cell subset. There is also evidence that both CD4 plus and CD8 plus T cells can suppress immune responses, and this might operate through direct killing of APCS through Suppressive cytokines. For example, TGF beta negative regulation of signal transduction or via the idiotype network. Third point is large granular lymphocytes, in short LGLS. This group of lymphocytes has the capacity to recognize the surface changes that occur on a variety of tumor cells and virally infected cells. LGLS damage these target cells, but unlike TC cells, they are very effective at recognizing cells which lack or have lost their MSC molecules. This action is sometimes called natural killer cell activity. Now let's know about the auxiliary cells. Numerous other cells mediate inflammation, the main purpose of which is to attract leukocytes and the soluble mediators of immunity towards a site of infection. First is basophils and mast cells. These have granules containing a variety of mediators that produce inflammation in surrounding tissues. These mediators are released when the cells are triggered. They can also synthesize and secrete a number of mediators which control the development of immune reactions. Now let's know about basophils. Basophils are found in very small numbers in the circulation, accounting for less than 0.2% of leukocytes. They are functionally similar to mast cells but are mobile circulating cells. Now let's know about the mast cells. Mast cells lie close to blood vessels in all tissues, and some of their mediators act on cells in the vessel walls. There are two different kinds of mast cells: the mucosal mast cell associated with mucosa and the connective tissue mast cell. MMCs appear to depend on T cells for their proliferation, while the CTMs do not. Both types of mast cells can be seen under light microscopy using basic dyes. Mature blood basophils have randomly distributed granules surrounded by membranes. The granules in both basophils and mast cells contain heparin, leukotrienes, histamine, and eosinophil chemotactic factor of anaphylaxis. The stimulus The stimulus for mast cell or basophil